Hello everyone, my name is Anthony. Today I'll be presenting our clinical abstract titled Learning from Failure, explaining clinical ML model prediction errors. Our failures are as important as our successes, particularly in clinical machine learning models because clinicians need to know the limits of their models when they are applied to their patients, and modelers need to understand failure to build better models. Of note, there are two error etiologies of key concern in ML models for clinical risk stratification. They are origin of false negatives, which affects the sensitivity of the model, and origin of false positives, which affects the specificity of the model. Proposed 2 should be used on ML models applied to held out test set results, ideally after ML model construction and hyperparameter selection. The tool has the following essential steps. 1. Filtering of errors. 2. Dividing filtered errors according to false negatives and false positives. 3. Applying SHAP specifically to the errors. 4. Aggregating the global SHAP value explanations of errors grouped by features. 5. Ranking group features according to their aggregate SHAP values. 6. Calculating average feature values and count number of errors in each feature group. We tested our methodology on the clinical data of a patient cohort of n equals to 7,960 adult patients extracted from MIMIC-4 dataset. Input features include patient demographics, comorbids, vitus, lab results, chronic medications, and imaging data. Outcome of interest was ICU admission within 24 hours from start time of ED visit. We trained a variety of ML models on the training dataset. Error explanation tool was applied to erroneous results of best performing ML model. The tool was applied on GBoost, which had the best held out predictive performance. Despite so, GBoost had 262 prediction errors, 11.3% of test set, of which 143 are false negatives and 119 are false positives. In points 2 and 3, we show examples of how top features contributing to false negatives and false positives can be ranked. The associated error counts are represented in brackets. In this figure, we show the top features' average contribution to false negatives on the left and the error counts associated with each feature on the right. A feature ranked highly for its average contribution to error might not have the highest error count and vice versa. A similar figure could be plotted for false positives as shown here. Both figures could be plotted with the failure explanation tool. In conclusion, we demonstrated a SHAP-based tool to explain clinical ML model errors and failures. You can also find the tool on our GitHub. Thank you and see you at MLHC 2022.